Hey everyone, so this video is on recombinant DNA. It's part of our biotechnology unit. It's the lesson after restriction enzymes. So the main intention of this video is to understand how recombinant DNA is formed, to understand how we can then use recombinant DNA for gene cloning, and we're going to also discuss how scientists use antibiotics to check if this process has worked. So first of all, what is recombinant DNA? So it's a term used to describe DNA which contains DNA from two or more different strands. So it's an artificially constructed piece of DNA. And it often uses bacterial plasmids, which are these circular pieces of DNA found in bacteria, which can replicate very quickly on their own. You can see in the diagram here, we've got a plasmid, we've got a gene, we're combining them to make recombinant DNA. So the basic steps in recombinant DNA is that you grab bacteria with a plasmid and you can remove that plasmid. You then grab the desired gene that you want to place into that. So it could be the gene, for example, to make insulin. So that would also be removed. And then together when they're combined, we end up getting recombinant DNA plasmids. So in other words, it's got the normal genes that are in the plasmid plus the desired gene from another source. Again, just cutting back in this video, you can see here, there's our plasmid, our desired gene is shown in blue. And when we combine them together, we can see we've got recombinant DNA. So the process of how that works obviously relies on restriction enzymes. So we discussed last week, restriction enzymes had specific recognition sites. So we're going to pick a restriction enzyme that will help us cut out the gene that we want. So in this diagram here, we're going to use insulin as our example. If we want insulin as our desired gene, we're going to pick a restriction enzyme that will cut on either side of that gene. So we've got one that cuts, I guess we could take to the left, and one that cuts to the right. And that lets us remove that gene that we want. And you'll notice now that they've actually got sticky ends. That's really helpful for inserting it into other sections of DNA. Again, we need to use the same restriction enzyme to cut the plasmid as well as the desired gene. That means that they have complementary sticky ends. So it means they're going to match up quite nicely. So we use the same restriction enzymes. We cut our plasmid. Okay, and you can see that when we do that, we're going to have a plasmid that's sort of opened up with sticky ends. We're then going to have a combination where we put that with our desired gene. Okay, we use a special enzyme known as DNA ligase, which is sort of like, it's sometimes often referred to as DNA glue, but it helps stick DNA pieces together. And what you end up then with is recombinant DNA. And you can see here, it's got both the plasmid genes shown in yellow and the desired gene shown in the blue inserted into one plasmid. So what we've successfully done now is we've taken the gene we wanted and placed it into a plasmid with the help of restriction enzymes and DNA ligase. Now, we use this process to help us through gene cloning. So sometimes we want to get a particular copy of a gene replicated many times, and bacteria are actually very helpful for doing this because they undergo binary fission, which you remember from last year is the process of splitting in half. It happens very quickly. It means we can actually place one plasmid into an original bacteria, let them replicate, and we end up with many, many copies of that gene now being expressed. So here, for example, we place our plasmid into the bacteria. It undergoes binary fission over and over, and you can see that each of these bacteria now have that plasmid with the desired gene present. So we might then harvest whatever that gene is responsible for. So it's a lot to get our head around, but I'll hopefully use this example of insulin. This is a real life use of recombinant DNA. So we actually get insulin from bacteria, and the way we do that is we place a plasmid inside a bacteria that also has the insulin gene inside of it. So you can see here, here's our plasmid. This section here is our human insulin gene. Now from there, we let the bacteria grow. So we put them in a fermentation tank. They go through binary fission, and then we end up with a bacteria that all contain the plasmids that we want. We then extract the insulin. Because these bacteria have got the gene for insulin production, they're now making insulin. We then harvest that. We then refine it and use it as a medicine. So from the very start of the process, we've extracted the gene that we want, place it into a plasmid, and place that then into bacteria and let that grow, and then eventually we harvest. So that's an example of how we use recombinant DNA to then undergo a process of gene cloning to, I guess, harvest or grow a gene or replicate a gene many, many times. Now, the problem with recombinant DNA is we don't always know how successful the plasmid is. Sometimes it really works well and the desired gene fits in the plasmid. Other times it doesn't work so well. So scientists will often put other genes into the plasmid to check if it actually works. So sometimes along with, let's say, the insulin gene, they'll also put a gene that will make the bacteria antibiotic and have resistance to antibiotics. And so then what they do is they test that by placing the bacteria into some antibiotics, and if it grows, then we know that the bacteria must have the plasmid that's worked. Okay, so it's almost like placing something into the plasmid as an extra test, and then we test it with antibiotics. That's a really, really common practice. The second one is sometimes they'll put a gene that will actually make the plasmid or the bacteria grow, and that's known as GFP, or green fluorescent protein. And so obviously the bacteria that are glowing bright green must have the plasmid inside of them working, because they've then got the gene for glowing in the dark. So again, it's another gene that they add to test if the process works. This is a common question in exams about testing for plasmid uptake, or seeing if gene clonings actually work. So knowing about antibiotic resistance and knowing about the green fluorescent protein is quite important to make sure it also works. So in review, we use plasmids for recombinant DNA, and they're the circular piece of DNA found in bacteria. We place desired genes into those plasmids with the help of restriction enzymes, and we use our DNA ligase to stick it together. We then might use that recombinant DNA in gene cloning and placing inside of proteins. And the use of antibiotics or green fluorescent proteins is a good way to check if that process has actually worked. So it's a lot taken in one video, but it's quite an important process, and we'll cover, I guess, parts of this over the next few lessons as well. Thanks.